Ancient microbiomes uncovering microbial secrets from history. Recent advances in DNA sequencing have opened a new window onto the past, the ancient microbiome. Every human is home to trillions of microbes, bacteria, viruses, fungi, etc., that live on and inside the body, influencing health and diet. But how did our microbial companions live with us in antiquity? Ancient remains, mummies, teeth, bones, and even fossilized feces can harbor DNA from those long-ago microbes. As one review notes, modern genomic methods let us explore the complex microbial ecologies that inhabit our bodies in the past. In fact, researchers describe our microbiomes as a vast new microbial self that interacts with human biology in every era. By studying microbial DNA preserved in archaeological samples, scientists are uncovering how diets, diseases, and even social practices shape the microbes of our ancestors. This emerging field of paleomicrobiology is rewriting what we know about human history and health. Archaeological time capsules of microbes. Archaeological excavations around the world, shown an Egyptian dig site, yield ancient remains and sediments. These samples can preserve microbial DNA from long ago. Archaeological sites offer many time capsule specimens for ancient microbiome research. Mummified humans, especially those naturally preserved, Egyptian or Andean mummies, frozen bodies like Otzi the Iceman, often retain gut contents and tissues with microbial DNA. Dental calculus, hardened plaque on teeth, is another treasure. The calcified deposits trap oral bacteria, viruses, food particles, and even pigments. As anthropologist Christina Warrener explains, dental calculus is the single richest source of ancient DNA in the archaeological record, providing a direct window into ancient diets, diseases, and environment. Coprolites, fossilized feces, likewise preserve gut microbes and parasites. DNA extracted from coprolites can reveal what people were eating and which microbes lived in their intestines. Even soil, latrine sediments, and cave earth can hold traces of human-associated microbes. In short, any context that rapidly dries or mineralizes organic matter can lock in microbial remains for millennia. Key types of archaeological samples used in ancient microbiome research include mummies, dental calculus, coprolites. Mummies, natural or artificial mummification, can preserve not only human tissue, but also internal microbial communities. For example, DNA from 3,500-year-old Andean mummies showed abundant bacteriophages, viruses that infect gut bacteria, from families like Siphoviridae and Myoviridae, indicating that even viral DNA can survive in mummified gut contents. Other studies have pulled whole bacterial genomes, for example Clostridium perfringens, from the 5,300-year-old Tyrolean Iceman. Dental calculus, hardened plaque on teeth, encapsulates oral microbes and food debris. It has been used to track the rise of dairy farming by finding milk proteins and shifts in oral bacteria with diet. Sequencing calculus from humans and even Neanderthals allows direct comparison of oral microbiomes across time and populations. Coprolites, fossil feces. Human coprolites preserved in dry caves or latrines contain ancient gut microbes, parasites, and dietary DNA. A 7,000-year-old coprolite from Japan revealed not only bacteria but also virus DNA, for example herpes virus and adenovirus fragments, showing how viral and bacterial gut genomes co-evolved. Coprolite analyses have differentiated distinct prehistoric cultures. For instance, two pre-Columbian Puerto Rican societies, 1170 AD, were shown to have very different gut microbiomes and parasite loads, reflecting different staple foods, maize and fungi versus raw fish. In short, a wide range of contexts, mummies, dental calculus, coprolites, and sediments, can lock in microbial DNA for later study. In the laboratory, researchers extract and sequence DNA from ancient samples. High-throughput sequencing of degraded DNA allows reconstruction of past microbial communities. Studying ancient microbes relies on modern molecular tools adapted for old, fragile DNA. First, DNA is carefully extracted from the archaeological sample in sterile conditions to avoid contamination. Then high-throughput sequencing, NGS, or targeted amplification is used to read whatever short DNA fragments remain. These sequences are compared to modern reference databases to identify microbial species. Emerging methods such as metagenomic sequencing and proteomics, the analysis of preserved proteins, are now used to characterize ancient microbiomes. For example, a 2020 study applied ecological network analysis to thousands of DNA reads from ancient coprolites and dental calculus, identifying keystone gut bacteria, like short-chain fatty acid producers, and showing functional similarities to modern hunter-gatherer microbiomes. Careful authentication is key. Ancient DNA has characteristic damage patterns, like cytosine deamination, which help confirm its authenticity. The end result is a profile of the ancient microbial community, essentially a snapshot of the person's gut or mouth at the time of death. Fascinating findings from ancient microbiomes, diet and culture clues. Ancient microbiomes can reveal what people ate and how cultures differed. In the Puerto Rico study, for example, two neighboring societies around 1000 AD showed distinct gut microbiota. Fucoid coprolites contained maize and certain fungal DNA, Basidiomycetes, indicating a maize-rich diet, while saladoid samples contained DNA from fish parasites, implying heavy consumption of seafood. Similarly, dental calculus analyses have tracked the spread of dairying. Milk proteins preserved in plaque show when and where people first consumed animal milk, shedding light on the history of lactose tolerance. Pathogens and ancient diseases. Microbiome studies are rewriting the history of epidemics. For instance, analysis of 16th century victims in colonial Mexico identified Salmonella enterica DNA in their dental calculus and gut samples, solving the mystery of the deadly Coccolistli epidemic of 1545. The famous Tyrolean Iceman's stomach yielded the genome of Helicobacter pylori, linking his gut bug to ancient strains. More broadly, researchers have reconstructed full genomes of notorious ancient pathogens like Yersinia pestis, Mycobacterium leprae, and H. pylori from old samples. Microbiome evolution and diversity. Ancient samples show that human microbiomes have undergone significant changes. A landmark study of approximately 2,000-year-old North American coprolites found that many gut bacteria present in ancient humans are now extinct. Implications for modern health and history. Ancient microbiome research is more than an academic curiosity. By comparing past and present microbiomes, scientists hope to understand the rise of modern chronic diseases and even restore lost microbes. For example, knowing which gut bacteria vanished over time may guide therapies, prebiotics or probiotics, to reintroduce beneficial strains. Similarly, insights into how diet, sanitation, and antibiotics altered our microbial partners can help public health. Wariner emphasizes that understanding when and why our microbiomes changed, with agriculture, urbanization, industrialization, is key to tackling modern issues like obesity, allergies, and autoimmune diseases. In sum, the study of ancient microbiomes is revealing the microbial side of human history. By digging into the DNA of old bones, teeth, and poop, researchers are uncovering the diets, diseases, and environments of ancient peoples. This deep look at our microbial past not only satisfies historical curiosity, it may also guide future medicine by showing how to recover the beneficial microbes that our industrial lifestyles have depleted. Thanks for watching. Please like, share, and subscribe to the channel Microbes Masters for more insights into the microbial world.